So let's begin. Welcome everyone to the Friday noon talk. Today we have the pleasure of welcoming Dr. Sangkyu Lee. Sangkyu is a physicist at the Memorial Sloan Kettering Cancer Center in New York, New York. But we know Q from his time at McGill, so he did both his master's and Ph.D. at the medical physics unit at McGill. And his PhD work was on the prediction of radiation pneumonitis by integrating biomarkers and dosimetry data. And we were just discussing before, this was one of the first machine learning style projects in medical physics, at least that I'm aware of at McGill. After his PhD, he left Montreal and joined Memorial Sloan Kettering for a 4-year medical residency, physics residency program, including a research fellowship under Dr. Joe Deasy. And following his graduation from residency, he's now currently working as a physicist at MSK. So, Q, I will hand it over to you, and you can go ahead.
Thank you, Pete, for introduction, and thank you for the opportunity to speak at my home institution where I spent a lot of my youth. And sorry for not making in person, but I hope you guys are doing well, warm and healthy. So, yeah, I have no conflict of interest, and this is my source of funding. So, precision medicine has become a buzzword, but how does it apply to our field of medical physics? So, in this cartoon, we have a group of patients, and each patient has different radiosensitivity, which is marked by different colors. And what has been done traditionally is to apply a dose-response relationship, which was obtained from a population average, and apply this to the whole patient group to get a single dose constraint for every patient. Of course, there are exceptions, such as prior radiation and comorbidities, then we might adjust the dose constraint to each patient. So what precision radiation oncology can do is to use some kind of assay and a computer model to identify a patient subgroup with different sensitivity, and we can customize the dose constraint to these subgroups according to their predicted sensitivity. And for the subgroup with lower sensitivity, we can even do dose escalation to enhance tumor control. So this is the blueprint. So in order to do precision radiation oncology, we need to get a full picture of a patient and the treatment. So what can be in the picture? So these are the different data inputs that makes up a full picture. So traditionally, we have used those volume parameters and clinical data, and imaging features has been studied very, very actively in a field called radiogenomics, and biomarkers is the topic of today. And biomarkers, there's a huge diversity of biomarkers such as radiosensitivity assay or RNA expression, protein expression. But in this talk, I'll limit the discussion to DNA sequence. So radiogenomics is defined as the study of the influence of genetic variation on radiation response, according to West and Barnett. In this talk, I will limit the discussion on what radiation response is to the response of the normal tissues to therapeutic radiation. So in this talk, I won't talk about a tumor response, which is a whole different topic. So single nucleotide polymorphism, or SNP, is one type of genetic variation that refers to the variation of nucleotides in one location in a DNA sequence. And in this example, there are 5 patients, and this is showing a small segment of the DNA sequence. And you can see these 5 patients have the same sequence, except this location where 3 patients has the base G, while 2 patients have a base T. So the SNPs were identified by a huge international research project called 1,000 Genome Projects. So what they did was to take sequence different ethnic groups around the world and compare the sequence between them and to identify the sequence variations. And they have found 88 million SNPs across the genome so far. And SNPs is just one of different sequence variations. There are other types of variations such as copy number variation, insertion, deletion. But in this talk, I will only discuss the SNP or single nucleotide polymorphism. So genome-wide association study, or GWAS, is the association between the disease or any trait of interest with the sequence variations in terms of SNPs. So I will use the 5-patient example again. So in this small segment of DNA, we have 2 copies of genes, which can be coded by this DNA base pair sequence. And SNP at this location can be converted into a number in terms of how many minor alleles it has. So major or minor alleles are determined by the prevalence of a particular base in the population. So in this example, the allele T is rarer than the major allele G. So in this case, T becomes a minor allele, where G is a major allele. So what we do is simply count how many T's each patient has. So patient 3 has 2 T's. So the patient 3 is coded at 2, and patient 2 has only 1 T. So it's assigned to a value of 1. And the next step is simply to do association test between the number of minor alleles and the outcome of interest, from which we get the effect size and p-value. But there are 2 important statistical considerations we have to consider when we do GWAS. So statistical power is a problem when we do GWAS, because the power is limited by the number of large number of SNPs and relatively small sample size. And when we apply a typical p-value threshold of 0.05, we will end up with a bunch of false positives. And so what we do in GWAS community is to apply a much more stringent p-value threshold. And 5 times 10 to the minus 8 is a very popular p-value threshold. Apply this p-value threshold to the p-values across the genome to identify lead SNPs, which is the SNPs that are postulated to be relevant to the outcome. So we take the lead SNPs and further study those smaller group of SNPs for its biological relevance. And another consideration is how prevalent each allele is. And it's more difficult to detect a signal from rare SNPs because the statistical power is also limited by the prevalence of the SNP. So typically the SNPs with allele frequency over 5% is considered. And in this plot, it shows the distribution of p-values from an example GWAS study across the genome. So in this plot, the x-axis is the location of the SNP sorted from the chromosome 1 to chromosome 22, and y-axis is minus log of the p-value. So when you pay attention to the magnitude, I don't know, mouse is frozen. The magnitude of the y-axis shows how low the p-values can get from typical GWAS studies. By the way, feel free to interrupt if you don't understand anything or if you have any questions. So another consideration while doing GWAS is linked equilibrium, or LD. So LD refers to local correlation between SNPs, and the LD occurs because the neighboring SNPs are more likely to be inherited together, and the SNPs that are far away are likely to be broken apart by gene recombination. So for this reason, it's typical to search the vicinity of the lead SNPs for the presence of any causal genes. So in this example, the lead SNPs is located in the coding region, and the SNPs directly has biological consequence by changing the protein sequence. Whereas in this example below, the lead SNPs is located in non-coding region, but it doesn't necessarily mean that this SNP is false positive when it's correlated to another SNP in the coding region that we didn't genotype. But due to this correlation, this SNP tags the actual causal SNP. So this SNP has no biological role by itself, but it can be a good way to get to the SNPs that actually do the biological work. So the GWAS study, this is the largest GWAS study to date. It was just published in Nature. So what they did was to find the genetic variation that explains the variation in human height, and the sample size was quite impressive, 5.4 million. And the SNPs that discovered collectively explained 40% of the human variance. So let me go back to radiation oncology. So in the early days, until 2009 or 10, people believed that a small number of well-known radiosensitivity genes, such as ATM or XRCC1, TGF-beta, can drive different toxicities regardless of its anatomical sites. There has been many publications that claim the association between the SNPs within the well-known sensitivity genes to different toxicity outcomes. So this paper by Barnett et al. Tried to replicate these findings in the independent data, and they were not able to find any of the associations in their independent cohort, and the reason they explained was the lack of statistical power due to small sample size. Many published studies didn't have many patients, and another problem was the multiple testing was not corrected to control the statistical power. So both contributed to a lot of false positives in the literatures. So nowadays, people adopt a GWAS approach where we search across the genome without any hypothesis, and this was fueled by more affordable GWAS genotyping cost. And the multi-center collaboration has become more frequent to increase sample size and also enable independent validation. And Radiogenomics Consortium is a very good example. So what we learned from radiogenomics GWAS so far was that there doesn't seem to be any key driving genes or SNPs that causes any toxicities independent of a site, which means that normal tissue complications are expected to be polygenic. So polygenic means a disease is influenced by a large number of SNPs or genes, and the effect of individual SNP or genes are very small, but enlarged, they have a large compound effect. This is in contrast to Mendelian disease, where there is a single mutation that has a huge impact to the outcome. But this phenomenon seems to be very rare in radiation oncology. So how can machine learning help in radiogenomics? So machine learning is best for multifactorial decision making. It can make a decision from different factors, unlike humans. So it really reflects the polygenic nature of the disease. But in order to do that, we need to be very careful about reducing dimensionality of data to separate the noise from the actual signal. And machine learning model is judged by how well it can predict in unseen data, whereas the GWAS is the goal of GWAS is to replicate SNPs in individual in the independent cohort. So the goal of GWAS is different from machine learning, and in my opinion, machine learning is more geared towards clinical applications because it can tell us if the patient will get complication or not. So now I will talk about the machine learning methodology that was specifically developed to analyze GWAS data, and it was published in this paper, and the credit goes to Jung-Han Oh, who was my postdoc mentor. So it started with the expectation that if we just use univariate correlation p-values, many of the strongly correlated SNPs are false positive, but some others will be true positives, which is illustrated in this diagram where red dots represent true positive and the blue dots are false positive. So first thing we do is to apply a crude dimensionality reduction with a p-value of 0.01. So I get asked a lot from reviewers when I try to publish, why did you choose 0.01? Sounds arbitrary, right? But the reason that we chose this value was for a typical GWAS study that we have had with a typical sample size and the GWAS array size, it gives around 1,000 SNPs or less p-values that falls under the p-value threshold. So that gives us the number of SNPs that are okay to handle by machine learning methodology, machine learning methods. So it's purely by convenience. And then what we do with the reduced SNP data set is so-called preconditioning. So what preconditioning does is to regress the outcome, toxicity outcome with respect to the principal components of the input features. So in short, what it does is to denoise the outcomes and reduce any possible bias that might exist in outcome scoring. So there's a outcome transformation step. Then what we do next is to train a random forest model. So random forest, for those of you who's not familiar, is basically the ensemble of trees. So each tree makes the decision whether the patient will get sick or not based on this combination of features. And the model as a whole makes a decision by taking the average of the predictions across the forest by individual trees. So what's cool about random forest is that it can not only predict, but also can tell us how important each feature is in predicting the correct outcomes. So we can use the random forest to rank the SNPs based on its impact on prediction accuracies. So there are two layers of filtering false positives. One is the univariate correlation, and the second is random forest based filtering. So here the SNPs are ranked by the importance that I mentioned from the random forest model. And what we do is to remove the SNPs that are ranked lower and see how the validation prediction accuracy changes. And what we have observed, of course, regardless of the outcomes we analyzed, was that as we remove the lower importance SNPs, we see the increase in the prediction accuracies. And this tells us that the SNPs that are ranked lower might be it's more likely to be false positive. So this is the second layer of filtering that I was talking about. So we take the high importance SNPs that I mentioned, and then we search for the genes that are in vicinity of these SNPs. And so we end up with a list of genes which are curated by the model. And unlike SNPs, genes have biological interpretation, so it's much easier to tell a story of what this result means. So we have a list of genes that are curated by the model, and we ask the question, what do these genes have in common? So to answer the question, we query these genes to a biological knowledge database, and there are so many biological knowledge databases that are publicly available, and we can do two types of analysis. So first analysis is so-called gene set enrichment analysis. What it does is to find any significant overlap between our gene of interest and a group of genes that have common function or disease. And the second analysis we can do is so-called interactome analysis. So it basically finds the small subset of genes that are connected to each other, and it's based on the observations that the connected genes are more likely to have a common function. So now I'm going to talk about my personal work that I did in MSK in applying these machine learning methodologies to three different treatment outcomes. So the first study that I'm going to introduce was about predicting urinary toxicity after prostate cancer treatments. So the data set came from Mount Sinai. The patient was treated with brachy with or without external beam, and the data set came with some clinical variables and 700,000 sleep markers. Unfortunately, we're not able to get a detailed RT planning data except the tumor BED. And the urinary toxicity was assessed using patient-reported outcome, patient-reported questionnaire at 2 years follow-up. So we trained a model for 4 different outcomes, which came from the IPSS questionnaires, and there are 4 symptom categories, and we made a model for each symptom category. And unfortunately, we had to take a subset of the patient with the baseline score of 0, because otherwise the outcome will be too much dependent on the baseline score. So we had to reduce our sample size. And in terms of input variables, there was no clinical variable that was selected by the initial filtering due to their weak association. So the models we built was purely made out of genomic variables. And the results so we tested the model in the holdout validation data set, and we found 2 out of 4 urinary symptoms were predicted with the AUC significantly better than random. And when we did biological analysis, it was very encouraging. So the picture on the left shows the biological processes that are significantly enriched with the list of genes that were discovered by the model. So each blob here represents one biological process, and the biological process were connected based on the similarities of the biological processes. So you can see different groups of biology, such as here, these purple dots represent processes related to neurogenesis, and this group over here represents the processes related to potassium ion transport, and these two biology groups have been associated with urinary tract function. So the previous the literature supports that the genes that we discovered from this modeling is actually relevant to our outcome of interest, which is urinary symptoms. And this plot, this diagram shows the group of genes that are connected to each other. And what we did was to systematic literature search to find any relevance between these genes and our outcomes, which is urinary symptoms, and the genes that were highlighted, underscored in red, have been previously reported to be associated with urinary symptoms. So the model that we built did not predict very well, but its biological relevance was very encouraging. So the second study that I want to introduce, at this time, we try to predict contralateral breast cancer after radiotherapy. And for those of you who may not know, contralateral breast cancer, or CBC, is a long-term risk from treating primary breast cancer. The prevalence is not very high, but because breast cancer patients survive very long, and collectively, it is a non-trivial public health risk. So, one of the postulated risk factor is the scatter radiation from whole breast radiotherapy, and the study has shown that a younger patient with age under 40 are much higher risk for contralateral breast cancer after receiving whole breast treatment. So we care study is a multi-center case control study to find genetic components of CBC, and fortunately we were able to get data from the WeCare study through some collaborators within the MSK. So the study cohort, again, we had to take a small subset of the WeCare study, which are known to be in higher risk than the rest of the group, which is the age of 40 and under, and contralateral dose, scatter dose over 1 gray. And again, we've tested the clinical variables as well, but the p-values from the clinical variables were not significant enough. So the model was again built using genomic data only. And the hold-up performance was AUC of 0.62. Again, this is better than random, but there's room to improve. And to be fair, the training sample size was very small. So it was kind of a stress test of how well our methodology can do in a small data. So again, the picture on the left shows the connected components of the genes that were found by the models. And we did a literature survey, and it was very impressive that these 8 genes that are connected are all relevant to breast cancer, and some of them were even associated with radiation-induced secondary cancer. And what we did on the right was that we compared the list of genes prioritized by our model versus other ranking methods, and the list of genes prioritized by our machine learning method led to more biological processes and more relevant biological processes compared to lasso univariate ranking methods. So once again, not the best prediction, but a very relevant biology to breast cancer. So the final project that I wanted to present here is, again, breast cancer outcome, but this time was fatigue. So fatigue is actually quite common to breast cancer patients after their treatments. And the prevalence rate was around 30%, according to this paper. And the biological mechanism of this fatigue was not fully understood. So the Kanto study was intended to answer this question by enrolling breast cancer patients across France. So this work was enabled by collaboration between us and the Institut Gustave Roussy in Paris, where the main PIs of this project works. So the Kanto study, the goal was to collect data to survey the quality of life of breast cancer patients, and fatigue especially was scored by a patient-reported questionnaire called EORTC-C30. So I trained 4 different models for each fatigue domains: overall, physical, emotional, and cognitive. Again, I had to take a subset of the cohort with a 0 baseline score due to, again, a strong dependence between the baseline score and the final outcome. And then we did the same univariate test to reduce the dimensionality. And in this time, there are many clinical variables that were strongly associated with the outcomes, such as anxiety, pain, depression, insomnia, which all make sense because fatigue, we all know that fatigue can be contributed by these factors. And so we built a model first with genomic variables only, which gave us 0.559 AUC in the holdout validation. And after we added these clinical variables that were significant, the AUC bumped up a little bit, but not too much. So this is something that we are working on to better integrate the clinical variables that are relevant to increase the prediction performance. Anyways, so we did the same scheme of biological interpretation of the model, and the figure on the left shows the connected components within the list of genes that were discovered by the model, and these two highlighted genes or cytokines that have been previously implicated in cancer-related fatigue. So it was, again, very encouraging result. And we also saw a significant enrichment between the list of genes from the model and the list of genes that were previously implicated in cognitive, mood, and depression disorders with a very strong p-value. So again, the model that we built was highly relevant to our outcome. So to summarize, what we learned from these exercises was our random forest model was worked quite well in highlighting the biology that's relevant to each outcomes that we analyze. And the biology tends to be very specific to the outcome of interest. For example, the biology that we found for urinary toxicity was relevant to the urinary tract, whereas for the breast cancer prediction, we found many breast cancer related genes. And so, but still, to be able to use our model clinically, we need to do better in improving predictive performance of our model. And it could be that we used only genomic features to predict while ignoring other factors. And it's expected that if we only consider genomics, we only see a small portion of the picture that I talked about, picture about the patient. So we are working on better integrating the clinical or dosimetric data to bump up the prediction AUC. And I'm also working on other radiotherapy outcomes, such as radionecrosis, which is a very challenging project because the treatment variables are very heterogeneous, and this is the outcome that has not been understood very well. People don't know the dose volumetric relationships, and there's seems to be a heterogeneity in dose response. So once we overcome the hurdle of data collection and harmonization, we'll be able to give some clinical guidance on which can be useful for planning brain radiosurgery. And data sharing for validation is important, but there are so many challenges that we have to overcome, such as cost, logistics, and privacy regulation is another hurdle. So for the project that I mentioned in France, they didn't grant us access to the data set online. So I had to travel to France to analyze the data, so which was great, but regulations like that can be a hurdle in freely sharing data for validation. And another issue that we have seen was, even though the data are collected and shared from different institutions, there is a problem with not having consistent protocols for outcome scoring or data collection. So it can be an issue when we compare toxicity in one institution versus another, and one institution is using one criteria while the other institution is using a different criteria, and we'll see the mismatch in the toxicity prevalence, and also the treatment method is also different between institutions. So that's where the consortium can really help in setting the protocols that harmonizes such as scoring and data collection schemes. So I'd like to acknowledge many people that contributed to this project, especially Dr. Zizi and Dr. O, and I'm happy to take questions.